Hey fellas, we are from Biology A 2024. We will learn about application of molecular biology and cell in our agricultural sector. So without any further ado, let's start. But before that, let us introduce ourselves. I am Daryll Alfaraby N with the last student number 002.
And my name is Siti Kavy with last digit name 044.
My name is Rahma Amanda with real last digit name 114.
My name is Oktavia Ramadhani, exit number 120.
Okay, introduction. Cell and molecular biology are branches of biology that study the structure, function, and mechanism of cell and molecular component of life, such as DNA, RNA, and protein. Rapid advances in this field have created significant opportunities across various sectors, including agriculture. In the agricultural content, cell and molecular biology play an important role in improving crop productivity, quality, and resilience. The application of technologies such as genetic engineering, tissue culture, and molecular analysis enable the development of superior crop varieties that are resistant to pests, disease, and environmental stresses. Along with the increasing demand for food due to population growth, the application of cell and molecular biology has become a strategic solution to support sustainable, efficient, and environmentally friendly agricultural practices. Okay, next. Next for urgency. This research was conducted because the production of plant with superior chain is very important to support the dependence of agriculture and various industries on plant researchers. Exciting traditional breeding methods require a lot of time and energy, so genome editing technology is needed to enable more efficient and rapid specific modification to plant DNA. And this is the journal.
Okay. Based on journal, role of molecular biology and cells. Cell and molecular biology plays an important role in understanding the basic mechanism of plant life at the genetic level, enable specific DNA modification to improve plant traits through genome editing technologies such as CRISPR or Cas9. This technology accelerates the plant improvement process in a more efficient manner compared to traditional methods. And explanations of one case study or application. One of the practical applications of cell and molecular biology in agriculture is the use of CRISPR or Cas9 technology to perform precision genome editing in plants. This technology operation utilize molecular mechanisms within the cell to precisely DNA at specific target genes, enable target and accurate genetic modifications. According to Waya et al. 2020, CRISPR or Cas9 technology has been widely applied in various agricultural crops to modify genes involved in disease resistance, tolerance to abiotic stress, and improvement in crop yield and quality. This system employs the Cas9 enzyme quite by acquired RNA to recognize and cleave specific DNA sequence within the plant genome. One example of its application is the knockout of genes that make plants susceptible to pathogen attacks. By inactivating these genes, plants can become more resistant to disease without disrupting their normal growth and development. Furthermore, editing genes involve in stress response pathway enable plants to exhibit better tolerance to extreme environment conditions such as drought and salinity.
Okay, the next what I will explain is main experience of CRISPR or Cas9 technology. The main experience of this technology lies in ability to process genetic modifications that are faster, more accurate, and more efficient compared to conventional breeding methods. Moreover, CRISPR or Cas9 enables the development of improved crop varieties without the insertion of foreign genes, making the resulting plant potentially more acceptable within modern agricultural systems. The application of CRISPR or Cas9 as described by Water et al. 2020 demonstrate that cell and molecular biology play a strategic role in supporting sustainable agriculture, particularly in addressing the challenge of increasing crop demand and climate change. Next, impacts or benefit of CRISPR and Cas9 application. The application of CRISPR or Cas9 technology in agriculture provides significant benefits, particularly in enhancing plant resistance to disease and pest, thereby reducing the reliance of chemical pesticides. The technology also enables crops to exhibit greater tolerance to abiotic stresses, such as drought and salinity. In addition, CRISPR or Cas9 contribute to improving crop yield and quality, including productivity, nutritional content, and other agronomic traits. Through a faster and more precise breeding process, the development of superior crop varieties can be accomplished in a shorter time compared to conventional methods, thereby supporting sustainable agriculture and food security. Water et al. 2020.
Alright, in this slide, I will explain you about the examples. As we can see on this slide, we can see several practical examples of how CRISPR's technology is applied in fruits and vegetables. And first, starting from the image on the left, this is lettuce. In lettuce, CRISPR has been used to improve traits such as a resistance to disease, tolerance to environmental stress, and in some cases, to reduce browning after harvest. These improvements help extend shelf life and maintain product quality, which is very important for leafy vegetables that are easily damaged during storage and transportation. Next, in the top of right image, we no, still there. Okay, and the next picture, you can see JB rich tomatoes, JB rich tomatoes using crisp PR, and then with these tomatoes, scientists modify genes involved in the GBA biosynthesis pathway so that the plant can naturally produce higher levels of GABA. This results in tomatoes with added functional value as GABA is known to support health. For example, by helping to regulate blood pressure and promoting relaxation without changing the appearance or taste of the fruit. And then in the bottom of the right image, we can see seedless watermelon. Through targeted gene editing, genes related to seed development can be modified, allowing the plant to produce fruits with few or no seeds. This makes the watermelon more convenient for consumers while maintaining its flavor, texture, and overall fruit quality. These examples show that CRISPR is not only used to increase crop yield, but also to improve quality, nutrition, and consumer convenience in modern agriculture. Next slide. So the conclusion is to conclude cell and molecular biology play a key role in the development of modern agriculture. CRISPR or Cas9 technology allows precise and efficient modification of plant genomes, making it possible to improve disease resistance, environmental stress tolerance, yield, and nutritional quality in targeted way. Importantly, many CRISPR Based improvements do not involve the insertion of variant genes, which may increase public acceptance compared to conventional genetic modification. Overall, CRISPR represents a powerful tool for supporting sustainable agriculture, strengthening food security, and addressing future challenges such as climate change. Thank you. All right, maybe is that enough from us about implementation of molecular biology and cell in agriculture sector. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.